Okay, good afternoon to everyone who is joining us today. Welcome to all school students, teachers, parents, and not forgetting all University Malaysia Sabah students and staff present in this webinar session. My name is Evelinda Christy Mojutan. I will be in charge as moderator for this session. We meet again for today's session of STEM Speaker Series. STEM Speaker Series is a collaboration between Science University Malaysia Sabah and MySTEM Ambassador University Malaysia Sabah. For those who join us for the first time, this STEM Speaker Series is getting full support from Sabah Ministry of Science, Technology and Innovation, which aim to deliver unique and interesting STEM topics designed by subject matter experts to increase the interest of school students, teachers, and community alike to venture in the fields of STEM. For any question for the audience today, please feel free to drop your question on the chat box at the right side of this YouTube live session. Please be informed that for today's session, your certificate will be given to school student and University Malaysia Sabah student. Therefore, I would like everyone to stay tuned with us to fill the form given through the link that will be shared at the end of the session. So now I would like to introduce our speaker. With us today is a distinguished academician from the Faculty of Science and Natural Resources, Dr. Lucky Goh Poh Wah. Little bit background of our speaker today. He has received his degree, master and PhD in biotechnology at University Malaysia Sabah in 2013, 2015, and 2019 respectively. His professional works includes a postdoctoral researcher in Faculty of Science and Natural Resources from July 2022 to January 2022. From July 2020 to January 2022. And now he is a senior lecturer in the same faculty, which is Faculty of Science and Natural Resources. His area of expertise are genetics, studying the genetic diversity and disease genes in rice using molecular methods, gut microbiome, and soil microorganisms study using next-generation sequencing, gene expression study using real-time PCR, and epidemiology and genetic polymorphisms study using molecular biology techniques such as PCR, RFLP, and single sequencing. Some of the awards and recognitions he has received are Postgraduate Inspiration Award at the Postgraduate Award Ceremony 2015 at University Malaysia Sabah, Abstract Awards at the International Congress of Blood and Marrow Transplantation 2020, and shortlisted for Young Scientist Award at the International Conference on Beneficial Microbes in 2018. So without further ado, please welcome Dr. Lucky with his topic entitled Genetics, the Code of Life.
Okay, thank you, moderator. Is my sound clear?
Yes, clear.
All right. So, good afternoon to everyone. So, first of all, I would like to thank you for taking your time to join this live sharing session. So, I would like to also thank the KSDI, okay, for supporting this event as well as supporting UMS all the way. So today I would like to just a little bit of sharing, all right? So with the topics of genetics, the code of life. So why this topic? So it is actually throughout the sharing, so you will see that actually we are very connected to each other, even though we are seems very different, right? So you can see that on the earth itself, okay, we have birds, animals, okay, so ranging down even to viruses, bacteria, fungi, the forest, the plant. So we may look very different, but we have one thing that unites us all. So that makes us to be comparable to each other. So that is our DNA. So the long name is deoxyribonucleic acid. So DNA is essentially, to me, I'm calling it the code of life because it actually links all together. So we are who we are and what we are. So mainly are due to our genetic. So that is the DNA itself. So what is genetics? What is DNA? So I believe that because of COVID, okay, many of us are knowing what is genetics, or what is DNA, or what is viruses, right? So I will just briefly go through. Okay, so this is a Watson-Crick double helix DNA model. So when we talk about DNA, a human DNA, all right? So essentially, it means that the double helix DNA model. So where is this double DNA helix model? Where can we find it? Okay, so it is basically inside our cell. So in a basic cell biology, right? So you can see that there is a cell, right? So there's a chromosome, all right? And then when we zoom into deeper and deeper, we can see that there is a DNA strand. So basically the DNA strands, regardless this is human, it is animal, bacteria, viruses, fungi, okay? So it is made up of essentially 4 bases. So A, T, C, and G. So adenine, thymine, guanine, and cytosine. So these are the 4 basic bases that what we are made up of in the cellular and even in the molecular level. So when we talk about genetics, okay, it is very difficult to run away from DNA. It is very difficult to run away from molecular genetics, so which is the DNA. That's why oftentimes when people talk about or discussing about genetics, so it will be going to and forth to molecular biology. So how big is the DNA? Where and what is DNA itself? So I'm just taking the human DNA, for example, right? So a human genome have 20,000 genes approximately, right? So it spans around 2 meter length, okay? So you can imagine that uh, a 2 meter string that can be compressed into a single cell, but you cannot observe the cell with your naked eye. So you need the microscope. So that is the level of complexity of the organization of our cells are. So the size, okay, is 3 gigabase pair. Okay, that 2 meter size is essentially a 3 gigabase pair. So it is from the Human Genome Project. Okay, so it's the first human genome project, so we can roughly estimate already what is the size of the human genome. So then from there on, as the technology improve and gets better, gets faster, gets easier, so it is now much more cheaper to sequence a human genome as compared to the earlier days. So that's why if we are facing, let's say, currently the COVID, so getting the COVID genetic data is very easy and very fast already, thankful to the technological advancement. So if to put it, give you a context of how big is 2 meter or how big is the 3 gigabase. So a complete A4 page, okay, can fit around 4,000 bases, okay, meaning that the ATCG bases that I mentioned previously. Okay, so one page can fit approximately 4,000 bases. So you imagine that your A4 paper, okay, you have one paper, right? So it's 4,000 bases. But now you have approximately 700,000 pages. So one ream of paper is I don't know, 50, 100, okay, pages. So you have so many reams of paper. So that is the information that is inside in every and single of our cell. So it's somehow studying genetics is very interesting in the context that how can our single cell store such a huge and big information, okay? So a DNA, okay, is like an instruction manual, okay? So if you are familiar with the IKEA furniture, okay, so I'm not promoting from them, okay? So the furniture comes with an instruction manual, right? So it tells you which board you need to assemble first, which screw going to which hole, so that you have a complete cupboard or a complete table, right? So IKEA provides you that instruction manual. So our DNA is the instruction manual. Our DNA is governing our cellular processes, whether we are breathing, how we speak, okay, all our psychomotor hand movements, leg movements, okay, our digestion, okay, our cellular division, okay. So all these processes are governing by the DNA itself. So it's basically a very big and huge instruction manual because it can govern a lot. Okay? So this is the central dogma. So I've touched a bit about molecular biology, okay, or molecular genetics. So oftentimes in molecular genetics, we are looking into the central dogma. So central dogma basically is how the information is transferred from the DNA to RNA and to protein. So that is through the transcription and translation process. So that's why the instruction manual is the DNA itself. The RNA is the intermediate, okay, is the messenger. And the proteins are the ones that is responsible for all the cellular processes. Whether we are tall, we are short, our eye color, hair color, that is the traits that we can observe. So traits that we could not observe are your cellular metabolism, your drug metabolism, right, your respiration, your Krebs cycle, okay? So those are actually governed by the proteins are the ones that is doing all the hard work, right? So DNA is simply the instruction manual on it. So if you think about the furniture example, so you have the instruction manual from the furniture company, which is the DNA. A complete cardboard would be the protein. So whether the protein or the cardboard is functional properly or it is not functional properly, okay, for example, whether if the DNA there is a damage, okay, so meaning that the instruction manual has a damage, so you do not have a complete and functional cupboard. So maybe the doors is stuck, the doors cannot be open, doors keep falling, okay, so that functionality, okay, is essentially the protein. So protein are the ones that is doing all the work, okay? So that's why in genetics, okay, it is essentially the code of life because it is a very important instruction manual to encodes for protein, okay, at the end of it. So the protein carry out all the cellular processes. So coming back to that, who are we? Okay, so essentially we are who we are. Okay, whether we are short, we are tall, we are healthy, we are sick, we are fat, we are thin, okay? So we are who we are. So all these traits or all these characteristics, right? So are essentially governed by the genes. Okay? So the DNA is the instruction manual. So, and then we have genes which are made up of DNA. Okay, so we have genes that is responsible for short and tall characteristics, drug metabolism, good or bad, okay, whether it's fit, fat or thin, right? So I believe that some of you might have encountered, like maybe in a family or within your friends group, okay, you have the same lifestyle. You eat the same thing. You exercise together. You essentially it's the same lifestyle, but somehow maybe you or your friends, okay, are very different. Okay? Even though both of you always eat McDonald's, okay, but one is easier to increase in weight, but another one, it still remains slim or thin, okay, or does not increase in weight. So that is essentially governing by the genes, governing by our DNA. That's why we are who we are. So coming back to this, the central dogma, okay, the DNA to RNA to protein. So that is the basic information, okay, of transfer from DNA to protein, and at the end of it, it gives the characteristics. So where do we get our DNA? Okay, it is inherited. Okay, so we inherit the sequences or the DNA sequence from our parents, right? So from our mother and father. So that's why even though we have siblings, right, but we are very different from our siblings because we are not twins, right? That's why twins are essentially identical copies of cells. Okay, so that's why when the cells are identically copied, so that's why they are twins. But if we and our siblings are not twins, so we do not look exactly 100% the same, right? They are still very different, right? Even though our parents are the same. So what all genetics is looking into is basically the inheritance, the passing of genetic information in the aspect of DNA from our parents to us. Okay? So that is what the main focus of genetics are looking into. Okay? But there are many other tools, okay, as well as many other applications of genetics, which I will go through later. So I will touch a bit about genetics in the unknown. Okay, what do I mean? Okay, genetics actually have been practiced actually. Okay, even though at that time, okay, genes and genetics, okay, have not been known yet. Okay, in the earlier days, maybe in the 1940s, 1950s. Okay, but it has been practiced. Okay, so this is a law, ancestral corn. Okay, the corn that we ate now, which is mostly a yellow color, is very big, very nice. Okay, very less hard, right? So, but this is a corn, okay, which is have not been undergo genetic selection, okay, meaning that this corn is actually very hard, okay, it's very hard to chew, okay, so on and so forth. So throughout the years, okay, so breeders, okay, or geneticists, okay, somehow they observe that when they breed the corn of small and juicy, or they breed the corn with the traits that they desire, okay, maybe it is big and pest resistance, okay, then when they breed these two corns, okay, the parental corn, so it will produce an offspring corn, right? So that offspring corn exhibit the characteristics from both parental corn, okay. It is like how we humans, okay, inherit the characteristics from our parents. So that's why one always argue or discussions, it keeps going on, are the corn that we are eating now genetically modified, so on and so forth, okay. So this is the corn that we ate now. So if you compare to the corn that has not gone through selective breeding, okay, has not gone through the genetic selection, so it is impossible to have the corn that we have now, okay? So that is what uh, genetics is doing, and genetics can be applied. So this is a broiler, so meaning that chicken lah, okay. So this figure illustrates that the body weight as well as the market age, okay, throughout the year. You can see that as the body weight increases across the year, okay, the market age reduces, meaning that younger broiler are getting heavier faster, right? So that is through genetics, through selective breeding, okay, which is why that we have uh, chickens that can grow very fast, okay, 3 months, 4 months, okay, then it can be marketed because it has reached the weight or the age, okay, that can be marketed, okay. So that's why from this we can see that selective breeding actually has already been going on. So once is something for you to think about, okay. So what are the mechanism of inheritance, okay, like across through the years, okay, what actually is causing the broiler or the corn, okay, to have a superior characteristics. So these are all based on the work of Mendels. So inheritance in terms of the genetics, when we talk about it, so essentially it refers to the very, very classical genetics. So it refers to Mendel's works of trait transmission. So it's 1822 until 1884. Okay, so we often refers to Mendel's. So in 1856, so Mendels observed that there is a pattern of inheritance, meaning that from the parental to the offspring, meaning that parental to the kids or parental to the younger generation. Okay, so he observed that there is a pattern. Okay, so then he intended to study what is the pattern of inheritance. So initially, he used mice, and then honeybees, and then finally he used peas, green peas, right? So these are the characteristics of pea plants that Mendel used, okay, in his inheritance experiments. So meaning that one parent can be round, another parent is wrinkled. One parent have the characteristics of yellow, another parent have the characteristics of green, okay, and so on and so forth. So these characteristics are not overlapping each other. If the plant is having a yellow seeds, so it will be yellow only. Okay? If the plant is green seeds, so the plant is green seeds only. Right? So that's why it is very easy to study genetics, okay, in terms of the inheritance during that time, okay, when there is no technology yet. Okay, so a simple walkthrough of Mendel's work. Okay, he crosses a short and tall plant. Okay, essentially, so it's a green pea plant. Okay, so this is what we refer to as the parental. So and then so it will produce an offspring. Right, we refer to the F1 generation. So when the F1 generation is crossed, so it will produce an offspring of F2. So Mendel's observed, okay, the ratio of tall to short are 3 to 1. Okay, so 3 tall, 1 short. So in fact, the actual numbers is 787 tall, so 277 short. So one when converted into ratio, so it will be approximately 3 to 1. So this is a pattern that Mendel's observed. Okay? So that's why we have all the theories of genetic inheritance or the Mendel's inheritance. Okay, so all these come from a very, very simple yet important experiment, which is from pea plants. So currently, how do we study genetics? How do we study the genome? What is the modern genetics, okay, which Mendel's pea plants has, of course, given us a very good information that there is a genetic trait transmission. So that is looking at the characteristics itself. Okay, so Mendel's can only look into the characteristics. So it at that time they could not look into what is the actual sequence of it, what is the DNA sequence of it. Right, so that is actually the gist of genetics. So currently we can study genetics or we can sequence the genome, okay, using sequencing. We have currently a Sanger sequencing, so that is a first generation sequencer, okay. So it is not a very long read length, okay, but it has a low error rate. So then we have a next generation sequencing. So by these two companies, okay. So they have different course specifications and capacity. Okay, but you can see that it has evolved, okay, from a machine that looking like this, that is without a touchscreen panel, somewhat a little bit. We still require a standalone PC to operate, to collect data, and move on towards now there's a touchscreen, so everything is on benchtop. So basically, you just need one instrument that's just sitting on the benchtop to do all the work to get all the genetics data, all the genetic sequences. So now currently we have the technology is moving on towards nanopore sequencing. So it's a third generation sequencer. So it is as big as roughly your standard thumb drive. Okay. So or roughly the size of your smartphones. Okay. So it is becoming very, very small. Okay. So from this first Sanger and then the next generation sequencer, which is big and require big space, huge consumption of electricity, okay, and moving on towards very, very small. Okay, so all this technology has allowed us to obtain the human genome or the plant genome, depending on what is the interest of the organism. So we can study genetics using knockout or knockdown, okay, meaning that we exclude a certain gene, okay, to see what is the function of the gene. Through RNA interference, as well as, I believe, gene editing like CRISPR, okay, is currently very popular and is very widely used currently. So in order to study, okay, the genome, in order to perform a knockout study, a knockdown, or CRISPR, okay, genome editing, okay, we need model organisms, okay. So model organisms are organisms that are being widely used, okay. Of course, because as I have gone through with Mendel just now, right? Initially he used mice and then bees, so he moved to pea plants, okay. So obviously mice is require a good place. You need to feed them, you need to take care of their well-being, okay, so on and so forth. And it is the life cycle is longer, okay, as compared to pea plants. Okay? So pea plants, the life cycle is shorter, so they can grow very fast. That's why studying genetics at that time, so using pea plants is somehow much more practical. But currently, we have different model organisms. Okay, we have a nematode. So this nematode is layman's term is worms lah, right? So it's a C. elegans. Okay, it is often used for neurological studies. Okay, so Arabidopsis. Okay, so it's a plant. Okay, a model organism for plants. So it's very often used in plant biology. Then we have zebrafish. Zebrafish is always being used for cancer research. So all these model organisms, okay, is much more practical. Okay, even though sometimes it may or may not reflect the actual situation. For example, we can study a gene of a human gene, okay, using the model organisms, okay. Like, let's say zebrafish, okay. But zebrafish, the organs are very different from human. The cellular metabolism are very different. The number of genes are different. The sequence of genes are totally different. The immune aspect of humans and zebrafish are very different. So sometimes zebrafish results may not represent what is happening in the human body, but we still need model organisms because it is impractical to conduct a study using human. Human lifespan or human life complete life cycle would be 80 years old, 90 years old. So if we were to conduct a study using humans, so you will need 70 years, then you'll get your results. Okay? So it's not practical. That's why model organisms, they have an advantage, which is they have a very short life cycle and it's very easy to manage. Then on top of that tree, so we have mouse, rats, okay, which is mostly on toxicology studies, cancer research as well. We have rabbit and sheep, okay, which is mostly for bone studies, okay, and fruit fly and so on and so forth. So there are many more. So what is the power of genetics? Okay, what can genetics give? Okay, what is the application of genetics? Okay, so I have touched a little bit, okay, previously. So now I will move into 4 aspects. Okay, so there are more than these 4. Okay, so I'm covering 4 aspects only. So first is the evolution. Okay, we can use genetics to study the evolution process, okay? Whether are humans really from Africa, and then the Africans migrate throughout the world and the different path. Okay, so to give like which is we are who we are now. Okay? As well as you can study the viruses. Okay, what is the evolution of viruses? Okay, so is virus A and virus B very different? Okay, so that is the first one. So, and genetics can be applied in pharmacogenetics. Okay, so it means that uh, a personalized medicine. Okay, so certain people respond to certain drug differently. For example, a cholesterol drug or diabetic drug can be very effective on individual A, but it is not effective on individual B. Right. So that's why a general practitioner that just give individual A and B the same drugs without knowing the genetics, okay, so would cause the patients to have oftentimes need to adjust the dosage of the drugs, change the type of medicine, okay, to a more suitable one, which where they are more responsive. So knowing genetics, knowing our DNA sequence, okay, general practitioner, pharmacist, okay, can have an somehow an idea of, okay, this individual is responsive to this drug, is not responsive to that drug. So I will just give the responsive drug or the responsive medicine. Okay? So that's why somehow sometimes you may notice that, or you may heard that, oh, these few people are taking the same type of medicine, same type of drugs, maybe cholesterol, high blood pressure drugs, okay, but somehow these require 2 tablets, these require 3 tablets, okay, so on and so forth. So that is due to the differences of our genetics. Because our genetics are different, so remember that our protein are different. So that's why all the drug metabolizing process are different, okay, between individuals or within different individuals. So forensics, okay, so genetics has been used in forensics for a very long time already, okay, so to really identify criminals or identify or do crime scene investigations, okay, because we have a very different genetics in terms of the genetic marker. So even our thumbprint, all of us are different, right? Unless you are twins, lah, okay. But somehow, sometimes twins might have different thumbprints. Okay, so our genetics are like our thumbprints. Okay, so even though we and our siblings we do not have the same thumbprints, so our genetics are also different. So because of that differences, okay, so forensics can use or can track the genetic marker and use it for criminal investigation. The fourth one is for diagnostics. Okay? Genetics can be used to diagnose diseases like inherited diseases such as hemophilia, thalassemia, Huntington disease, cystic fibrosis, so on and so forth. Many inherited disease. Besides inherited disease, genetics can also be used to diagnose infectious disease. So some people have wounds that cannot be healed, right? So then doctors would conduct a microbiological study to look into the genetics, right? So I touched a little bit about evolution. So now I will move on a little bit more. Okay? This is the COVID virus, okay, phylogenetic tree. So the orange color essentially is the Omicron. Okay, so all the individual dots are actually COVID viruses isolate from different individuals. Through genetics, we can know their genetic difference, okay, and group them. Okay, that's why we have what Delta, Omicron, Alpha, Gamma, so on and so forth. Okay, all these many different type of COVID strains. So in fact, we can even plot to see which country has what strain, what is the distribution, or what is the proportion of COVID strains in each country, as well as we can look into what is the diversity of the genes inside a COVID virus in between strain A or strain B. Yes, they can be categorized as the same Omicron, but they sometimes have genetic differences. So genetic data, molecular genetic data, okay, is quantifiable. That's why we can know how different they are from one isolate to another, from one strain to another strain. So pharmacogenetics, okay, terms for medicine and drugs, okay. So because the DNA is essentially the instruction manual, at the end of it, it will encode for a protein. So that protein is carrying out all the drug metabolizing functions. So this is a study that shows that having different genetic marker will have a different response as well as different adverse drug reactions if they are given a certain drug. Okay? So that is how pharmacogenetics can be used. So forensics, okay? So I've touched, so forensics are using DNA fingerprinting, okay? So to compare the marker in between individuals or in between the blood samples that they collect at the crime scene, and they compare it to the blood samples of individuals to see how similar or how different they are to proceed on to their criminal investigations. So there is actually a system, okay, a CODIS system, okay, a combined DNA indexing system. So it is essentially looking into different, I think 13 genetic markers, okay, to identify an individuals for criminal investigations. So that 13 genetic markers are essentially spanned on different parts of our DNA. That's why we always go back to knowing the sequence of DNA, because without knowing the sequence of DNA, all the genetic markers, okay, cannot be identified. Okay, so that's why the very basic of genetics is to know the sequence of the DNA. So in diagnostics for bacterial identification, okay, So patients who are in sepsis, okay, which is a clinical condition, okay, that is caused by a bacterial infection, right? So clinician would perform a culture to see what is the species or what is the bacteria that is infecting the wound or circulating in the bloodstream. Okay? A normal microbiology culture would take time, would take sometimes 1 week, okay, or maybe 3 days, but that can be overcome by using genetics because in hospitals, so diagnostics have to be made fast because it is a matter of life and death. So by using genetics, it can be done within 1 day or the slowest maybe 2 days, okay, to identify what is the strain of the bacteria that caused the diseases. So another one is to diagnose COVID virus. So I believe all of us have heard a lot about it. So whether RT-PCR, okay, so on and so forth lah. Okay, so then human diseases. So Huntington disease, okay, is inheritance disease. So on the left, you can see that it is a brain slide, okay, of 48 years old, and then a right is a normal brain, which is at 34 years old. So you can see there's a thinning of the cortex, okay, the shrinkage of the brain, okay, whether it's a gray, white matter, the changes. Okay? So Huntington disease is a neurological disorder that is caused by HTT gene. So this is actually even though we talk genetics, okay, oftentimes we say about human, the aspects of humans, but genetics can be applied for animal diseases as well. So in the end, having inside the field of genetics for very long, okay, so when we talk about genetics, or it is a, a one of a subfield of biotech, right? So when people heard the word biotech, it is often we do things in our own labs, we do things on our own bench, okay, it is always indoors, we do not have friends, okay, so on and so forth. But actually, in my life, okay, until now, so it is not always being on my own, okay. So of course, I gave trainings to people, okay. So that is how we learn, okay, as well as when we train other people, okay. So biotech is not really, or genetics is not really staying inside the lab, okay. And then we have a lot of activities within our faculty, okay. I'm in the Faculty of Science and Natural Resources, so we have students activities. I think this is one of the time where we have our barbecue sessions, okay. So and this is the time where we have our buka puasa session, okay, with all the peoples, okay, that I met, okay, all the organizing teams, all the members. This is where I go to the field sampling with the students. Okay, of course there's one, but in fact there's many more lah. So we go to the field under the sun, we go to the mangrove forest, we go to the open sea, okay, many places that we go, okay. So we go with my colleagues and friends, okay, we go to conferences, we go to traveling, okay, we go to talk with other people, exchange ideas, okay. Of course, more student activities, okay. This is, I think, the most recent one, okay, in the last few days, okay, in our faculty. So, and this is one of a photo of me in surgical room, okay, in the operation theater, working with the doctors, okay, when they are working on the patients. So we have some collaborative work with the hospital doctors where we need to change into hospital clothes, hospital scrubs, okay. It's very eye-opening, okay. And then this is one of our, also I think it's during the puasa month, the fasting month, okay, where we distribute foods to the students, okay, and of course these are my very close students where I teach and guide them in the lab, okay. So if they have problems, then we sit down, we discuss, okay. So all in all, in the field of genetics, in the field of biotech, yes, we spend a lot of time in the lab, no doubt. We spend a lot of time alone, okay, thinking of how we want to solve the problems we face in the experiments, why the experiment fails. Uh, but at the same time, we can also go to the field work, mingle around with friends, okay, with colleagues, okay. So all in all, it is still an ongoing journey. So me myself, I look forward to it. Okay. So, and that's all for me today. And thank you for your time. Okay, and thank you as well for the organizing committee for inviting me to give this session.
Okay, it was a very interesting and wonderful topic and very well presented as well. Now we will open for Q&A session. If you have any question, please feel free to drop your question on the chat box. Okay, I think there are no questions, Doctor.
Okay.
Okay, we hope if there's no question, then we will close this Q&A session. And we hope the audience enjoyed this amazing and interesting presentation by our wonderful speaker. The link for registration and e-certificate is now being shared for school student and UMS student. Please fill in your details and answer the simple feedback questionnaire. All students will receive e-certificate while SDP point will be given to UMS student only. So thank you again, Doctor, for the time shared with us. And thank you also to all the audience for your participation. We look forward to seeing you in the next 10 speaker series. It was wonderful and a pleasure to have all of you with us. With this, we conclude our webinar. Thank you for the cooperation and see you again.